So in this lecture, what we're going to be going through is the neuronal membrane at rest. So in chapter 2, we talked about neuroanatomy in the case, or neuronal physiology, in regards to the shape of the neuron, the different parts of the neuron, and what's essentially the shape of the neuron and how things go in terms of functioning. This lecture, we're going to focus more so on what's happening to the neuronal membrane at rest, where are ions at, and a brief introduction into what the action potential is. We'll get into some ideas of proteins in terms of receptors and transporters and pumps, and really start to look at the chemical components of neuronal communication. So let's start. So the nervous system collects, distributes, and integrates information together. So this is what happens in the neuron. And it does this through using electrical signals that sweep down the axon. Now, this is not like a telephone wire where you have free electrons. The electrical charge is in the cytosol of the axon and it's carried by ions. And ions are charged atoms. So in this class, you're going to have very important ions to remember are sodium, which is Na+, potassium, which is K+, calcium, which is Ca2+, chloride, which is Cl-, -1. And neurons are bathed in extracellular fluid, which are composed of Na+ and Cl- ions, or sodium and chloride ions. Now, the action potential is known as the nerve impulse. And a potential in this context is the separation of electrical charge that is seen across the membrane. So if you recall from chapter 2, we have the neuronal membrane, which is separating the inside of the neuron from the outside, right? So all the stuff inside the neuron is able to stay within the neuron due to the fact that there's a neuronal membrane. So the action potential is a non-decremental fixed signal size and duration signal, whereas a resting membrane potential is what is going on when the neuron is not active in terms of sending signals. And the frequency of the action potential encodes information. So this lecture is going to be really important for setting up our discussion on the action potential, which is going to happen next class or next video. So cells are capable of generating and conducting action potentials, and they are said to have an excitable membrane. So at rest, the cytosol that is inside the membrane surface has a negative electrical charge compared to the outside. And this is a very important number to remember for this class. The resting membrane potential of an average neuron is around -70 millivolts. That just means that compared to the outside, if you were to measure the potential inside of a neuron, it is about -70 millivolts. So during an action potential, which is when a signal is propagated and sent down the axon, you have a reversal of this potential where you have about -70 millivolts here at the resting potential, where it actually for a short period of time gets so depolarized that you're completely positive. And then the neuronal membrane is repolarized and goes down to threshold again. And we'll talk about each of these segments of the action potential in due time. So what is going on in the cytosol and the extracellular fluid? So water is a key component. This is the key ingredient in the intracellular and extracellular fluid. Ions that are dissolved in the water, and they are responsible for the resting and action potentials. So remember that ions are charged particles, and so we have sodium and chloride ions, we have potassium ions and calcium ions. So a key feature of water is that it's a polar solvent. So it's held together by polar covalent bonds. There's an uneven distribution of charge here. So this water molecule has a dipole moment. There is an unequal distribution of charge. So oxygen carries a net negative charge. Hydrogen carries a net positive charge. So ions are atoms or molecules with a net electrical charge. Cations carry a positive charge. Anions carry a negative charge. So table salt. Table salt is an ionic compound. It's held together through ionic bonds and attracted to other oppositely charged molecules. And this is the idea of positive and negatives are attracted to each other. So spheres of hydration are clouds of water surrounding ions that help to insulate them from each other. So these hydration shells are seen right here. So here's a water molecule right here with its oxygen ion is more attracted to the sodium ion here. And then you have your hydrogen ions on the other side. Around a chloride ion, you see that there's more attraction where the hydrogen is attracted to the chloride versus the oxygen. Now these spheres of hydration are going to come into play when we talk about pores for or channels for sodium versus potassium, and why sodium and potassium can't just equally go through each other's pores. So important ions to note for this class right here, and what I'll do is I'll just change this pen color, I guess, to green. These are going to be very important for you to understand and know as we go throughout the course. So later on, when I talk about sodium or potassium and I write Na plus or K plus, I'm not going to have to define that for you. So the phospholipid membrane helps to isolate the cytosol, which is the intracellular fluid, from the extracellular fluid out here. So hydrophilic compounds, or so stands for water-loving compounds, dissolve in water due to uneven electrical charges, whereas hydrophobic compounds like oil do not dissolve in water due to even electrical charges. So lipids are examples of a hydrophobic species that's water-fearing. Now, the composition of the phospholipid membrane helps to contribute to the resting and action potentials due to the fact that it's not allowing everything to be able to actually go through. You need channels to allow for ions to actually flow through. So here's the phospholipid bilayer. You have phosphate heads, so hydrophilic heads, and then you have lipid or hydrophobic tails. This is going to allow for select things to be able to pass through. In order to get from the outside to the inside and vice versa, you're actually going to require there be a channel. So this goes back to our discussion on proteins. So proteins are chains of amino acids that are folded in a particular order. So the types and distribution of proteins helps to distinguish neurons from non-neuronal cells. Remember that all the organs in our body, all our cells in our body have the same DNA, but it depends on what genes or segments of DNA are going to be transcribed and translated through the process of transcription and translation, respectively. So molecules assembled from amino acids, and these are composed of enzymes, cytoskeletal elements, and receptors, as well as transporters. So special proteins span the phospholipid bilayer. Remember that here is the phospholipid bilayer. In order to get from the outside to the inside, extracellular, intracellular, you're going to need something like a channel. Something that will go across this membrane right here, allowing for something like a charged species, which normally wouldn't be able to pass through the phospholipid bilayer, allowing for it to cross through. So this provides routes for ions to cross the membrane, and it helps to control the resting and action potentials. And this will be a lot more transparent and a lot more clear when we start talking about the action potential. So the protein structure. So here is your basic amino acid structure. You have your carboxylic acid functional group and your amino group. Remember from last video, we talked about how an amino acid is just that. It contains an amino and an acid group. It has a hydrogen off of the alpha carbon, and then this R group is what gives each amino acid its particular chemical reactivity. So the size and nature of the R group helps to determine its chemical relationship and reactivity with other species and the way that it can fold. So you have amino acids that have very hydrophobic R groups, such as valine, leucine, isoleucine, phenylalanine. Phenylalanine also has a benzene or an aromatic ring, and methionine. You have amino acids with hydrophilic R groups, aspartic acid, glutamic acid. These are also known as aspartate and glutamate, asparagine and glutamine, lysine, arginine, and histidine. And then you have other amino acids. Glycine is the only non-chiral amino acid. Alanine, cysteine, serine, threonine, tyrosine, proline, and tryptophan. Now, what's important to understand about these chemical R groups is that they are going to impact the way that a certain protein will fold in and on itself. So certain things will be attracted. So if we're talking about a phospholipid bilayer, you might expect in a protein that is going across this membrane that the amino acids in the hydrophobic side will behave a certain way and will be attracted in a certain way compared to those that are on the extracellular or intracellular sides. There are certain amino acids with charges, and these charges might be able to attract positive or negative ions. And then, so if that's the case, then you might actually have mutations that can result in loss of transport or loss of the ability of ions to flow through certain channels. So proteins are synthesized by ribosomes. So again, going back to the idea of whether we are on a free-floating ribosome, which would give rise to an enzyme, or a ribosome that is bound to the endoplasmic reticulum, giving us a membrane-bound protein. Amino acids are connected by bonds known as peptide bonds, and these join the amino and carboxyl groups of two amino acids. So here's the carboxyl group and the amino group of two amino acids, and this bond right here that is connecting these two amino acids is known as a peptide bond. And you'll see in a peptide, or in a small protein, you have multiple peptide bonds. Again, connecting each of these amino acids together. And we read this from the N-terminus to the carboxyl, or the C-terminus. So a protein made up of a single chain of amino acids is known as a polypeptide. Now there are 4 levels of protein structure. Sorry, if I ever have a brief pause, I'm usually just grabbing a drink of water. So the 4 levels of protein structure are primary, secondary, tertiary, and quaternary structure. The primary level of protein structure is the actual amino acids that make up the peptide. So this is the most basic that you can get. If you're trying to understand what a protein is, the primary level of its structure would be, well, what amino acids actually make it up? The secondary level of protein structure involves the local spatial organization and arrangement of the peptide backbone. So you can easily localize these structural elements. You can look for alpha helices and beta sheets. So the way that this peptide backbone will actually arrange in space. So as we have all these amino acids together, they're going to arrange in particular ways, either in helices or in sheets. The tertiary structure is the comprehensive 3D structure that a protein will take in a single polypeptide chain. So the tertiary structure of a protein is its own single polypeptide chain, the way that it folds in and on itself. So there's going to be particular interactions between the R groups of particular amino acids. And then the quaternary structure is when these different polypeptide chains all interact together to form a larger molecule, like a receptor. So subunits are the different polypeptides that contribute to a protein with quaternary structure. So a large protein like a receptor has a quaternary structure made up of several different subunits. So changes in different levels here, so primary, secondary, tertiary, can have drastic effects on other levels. So if you think about this, how is this so? And I'll give you a brief second to think about that. So let's say that you change the primary structure. Let's say we take this phenylalanine and we take it out, right? If we take out that phenylalanine, that's going to impact the way that glycine and glutamate are now interacting with each other. That's going to impact the way that this protein might interact with some other amino acid when it's folding on itself. That's going to drastically affect the secondary and the tertiary structures. Let's say the secondary structure could be definitely affected if you were to take something like a glycine out and put a proline. Prolines are known to cause kinks in helices. So that is going to impact the secondary structure, which can then impact the way that the tertiary structure occurs. So changing different levels, and particularly just changing the primary structure of the amino acid or changing the amino acids that are making up this protein are going to have drastic effects on the way that the protein folds in and around itself, and then also works together with other subunits. So here's our protein structure. We have our individual amino acids. These are going to arrange locally in a spatial backbone, so in helices or sheets. These will go into a tertiary structure around the single polypeptide and how it interacts in itself, and then that's known as a subunit, and then multiple subunits can come together to form a quaternary structure, something like a transporter or a receptor. So channel proteins are what help to allow for ions to actually be able to come into the cell, into and or out of. So polar R groups, so R groups that are going to be attracted to water, tend to be exposed to the watery environments in and out of the cell. Nonpolar R groups tend to be within the membrane. So right here, if we had a membrane channel that's right here, you would have more nonpolar R groups lining inside, and then you would have more polar R groups on the outside. So channel composition varies between whatever kind of protein you're looking at. So a sodium channel or a particular receptor for a particular neurotransmitter will vary based on its own composition, and this helps to determine the different properties. So membrane-spanning proteins always have 4 to 6 subunits, and these different subunits are going to come together in a quaternary structure to provide the channel with its own composition. So ion selectivity is specified by the diameter of the pore and the nature of the R groups that line it. So basically what this is saying is that each receptor is going to be selective for what ions it allows to come into the cell or out of the cell, based on the fact that, well, if the pore is too small, if an ion has a very large diameter, it can't go through. Also, if you're trying to have, let's say, sodium flow in, which has a positive charge, and you have something like a mutation that causes a lot of positive amino acids to line the pore, this is not going to work because it's going to be repulsed. There's going to be an electrical repulsion. Gating is also another critical factor for channel proteins, in that opening and closing of channels due to changes in the local microenvironment can allow for ions to pass through and or leave. So if the gate is closed, physically nothing is going to be able to go through, but if the channel is open, then that allows for transport of ions in or out. Another type of protein is a pump, and this is formed by membrane-spanning proteins. Now, what's different from a regular channel protein versus a pump is what we'll talk about in a little bit when we talk about concentration gradients. And if we have things that go against their concentration gradient, you need energy from ATP. So ATP is produced in the mitochondria, and this is cellular energy. And in neuronal signaling, what happens is we need these ional pumps to be able to take sodium and potassium against their concentration gradient. So if you think about two rooms with a door connecting the rooms, and you have 100 cats on one side and 0 cats on the other, if you were to open the door, where would the cats go? The cats would go into the empty room, right? They wouldn't stay in the crowded room. Now, there would be an eventual movement of cats down this way, where eventually you'd have roughly 50-50. You would reach an equilibrium of cats. Now, if you wanted to move extra cats from, let's say, this room now had 20 cats, and you wanted to move it into a room of 80 cats, you would have to use energy. This is the same basic idea. Sodium is going to want to flow into the cell due to the fact that it is very high on the outside versus the inside. But if you want to take sodium, which is higher on the outside, if you want to take it from the inside to the outside, you have to go against the way that it normally would. Therefore, you have to use energy through ATP. So, for the rest of this lecture, what we're going to be doing is talking about movement of ions. So diffusion, concentration gradients, and we'll start talking about the idea of conductance and resistance and current. So the movement of ions is influenced by two different factors, diffusion and electricity. So diffusion is what we refer to as a concentration gradient. So ions that are dissolved in a solution will tend to distribute evenly. You're not going to have, in a glass of salt water, certain sips that are not salty and certain sips that are completely salty. The dissolved ions are going to distribute evenly. Now, ions flow down concentration gradients in two cases. When channels are permeable, so when you actually have channels, like doorways, and if a concentration gradient exists. So here in this first box right here, in figure A, there are physically no channels. So this is a makeshift membrane right here. You have a dissolved amount of sodium and chloride ions, but if there's no channel for sodium, then it cannot go through, right? If you were in a room and there was no door, you could not go through. Now, if you stick some channels in the membrane, there is now a way for the ions to flow. The ions have a door, they can leave. And remember, just like the idea with the cats, if there's a very high amount of sodium on one side and there's none on the other side, and you give sodium the chance to flow, sodium is going to go down to the area of least concentration, or less concentration. It's not going to willingly stay here. Now, in the absence of any other factors, movement of these ions is going to cease when there is an equal distribution. So there's an equal distribution on the left and the right. There's not necessarily going to be any movement. Now, if there's a different charge distribution, there could be electrostatic gradients, etc., but we'll get in that in due time. So the idea of diffusion and concentration gradients. If you got that, good. If not, just rewind this and watch it again and see if it can make a little bit more sense. Now, electrical gradients and electricity. So remember that ions are electrically charged. Sodium Na+, chloride Cl-. Electrical current, or the movement of electrical charge, influences the way that ions move. And this is measured in amps, or amperes. So remember that opposite charges attract and like charges repel. So sodium is a cation and it will flow toward the cathode. Chloride is an anion and will flow toward the anode. Cl carries a negative charge and will flow towards the positive. Sodium is positive and will flow towards the negative. So, electrical conductance and potential will help to determine how much current will flow in a given system. So, electrical conductance is denoted G, measured in Siemens, and resistance, which is measured in ohms. Now, conductance is the relative ability of an electrical charge to be able to move or migrate from one point to another. So this is going to depend on several things. So it's going to depend on the number of ions or electrons that are available to carry the charge and the ease by which these can actually pass through space. R is resistance, and this is the relative inability of an electrical charge to migrate. So the formula for R, or resistance, is 1 divided by conductance, or G. So the resistance is the inverse of conductance. So conductance is the ability of an electrical charge to actually move from one place to another, and resistance, like the word it's based on, is the inability of an electrical charge to migrate. Now, electrical potential is measured in volts, and this is voltage. This is the force exerted on a particular charged molecule, and this reflects the difference in charges between the anode and the cathode. The electrical current flows across a membrane. Now, voltage differences between the intracellular and extracellular space results in a resting membrane potential of negative 70 millivolts. If one side of the membrane possesses a relatively negative electrical charge, then positively charged ions are going to be attracted to that side because opposite charges attract. So let me just say that one more time. If one side of the membrane, so let's say the inside of the membrane, has a relatively negative charge, then positively charged ions are going to be attracted to it. Now, this is if the membrane possesses channels that are permeable to that ion. So if you have electrical conductance. And 2, if there is an electrical potential across the membrane. So is there a difference in electrical charge that is going to allow for current to flow? And does the membrane actually have a door whereby your ions can flow through? And doors in this case would be an example of a channel protein. Okay, now according to Ohm's law, your current is going to be the product of the conductance, measured in Siemens, multiplied by volts, or voltage, in the potential difference. So look at figure A, no current flows since conductance is 0. So current equals 0 times voltage, right? 0 times V. Anything multiplied by 0 is 0. So if conductance is 0, no current is going to flow, even when there's a large potential difference, right? V can be as big as you want it to be, but if you physically have no means by which these charges can flow, then your current will always be 0. Now, the potential difference, if the potential difference is 0, no current is going to flow even if the conductance is large. So here we go, we have our electrical current. Ions can now cross. The current flows in the direction of cation movement, so sodium is a cation, and the current will flow in this direction. Current is defined as the positive in the direction of positive, as positive charge movement. So driving an ion across the membrane electrically requires there be some conductance, meaning that the membrane has some channels permeable to the ions, and that an electrical potential difference exists across that membrane. So both of these values should be something other than 0, right? In order for current to flow. Okay. So the membrane potential is the voltage across the neuronal membrane at any given moment. This is abbreviated V sub m. The membrane potential is measured through inserting a tiny microelectrode into the cytosol. The voltmeter will measure the electrical potential difference between the tip of the microelectrode that is within the cell and the wire that's often in the extracellular fluid outside the cell. Now, steady difference, the resting potential is maintained whenever the neuron is not generating impulses. Inside of the cell will be electrically negative compared to the outside. The resting membrane potential is about negative 65 millivolts. Some other papers say negative 70. In this class, negative 65 and negative 70 are always going to be the answers. I'm never going to put on a test or a quiz negative 65 and negative 70 as options because it depends on which paper you read, honestly. Some will say negative 65, some will say negative 70. Now, why is this the case? So why is there more of a negative charge inside the cell? Well, the ions that are available, where they're distributed, what channels are available and open, these are all going to play a part in this resting membrane potential, but also you have more sodium chloride on the outside, more potassium inside, and what really drives this in the negative way is you have a very large number of proteins that carry negative charges that are found within inside the cell. Let me grab some water. So now we're going to discuss equilibrium potentials. An ionic equilibrium potential is the electrical potential difference that will exactly balance an ionic concentration gradient. Basically what this is saying is at what point or at what potential is there going to be a balance between charge and concentration gradient. So K plus, or potassium, is 20 times more concentrated on the inside of the cell than on the outside of the cell. So there's a large concentration difference, but electrically neutral. When you introduce channels, ions are going to be able to move down their concentration gradient. Again, in this case, we are only, let me grab a different color on the pen. Here, you only have potassium channels. So the conductance of potassium is going to be greater than 0. You're allowing for potassium to flow through. Now, as more and more potassium flows through, you have more and more negative building up on the left side, more and more positive building on the right side. The electrical potential will eventually balance out the concentration gradient and no more movement of potassium will occur. Now, in this case, you're only allowing for potassium to flow through. The equilibrium potential for potassium is negative 80 millivolts. At negative 80 millivolts, there's going to be no more movement of potassium across the membrane, as the charge is going to be equal. So the electrical potential is going to balance out the concentration gradient. So a net accumulation of positive charge on the outside and negative charge on the inside retards the movement of positively charged ions from the inside to the out. So in this case, an equilibrium is established. So there is no net movement of ions across the membrane, leaving a charge difference between the two sides. So all that's required to generate a steady electrical potential difference is an ionic concentration gradient and a permeability to a selective ion. So again, going back to the idea that there needs to actually be permeability, you need a channel, and you need there to be a concentration gradient. So there are 4 important points. So large changes in the membrane potential can be caused by minuscule changes in ionic concentrations. The net difference in electrical charge needs to be taken into account inside and outside of the membrane surface. And capacitance is the fact that the membrane stores this electrical charge. The rate of movement of ions across the membrane is driven at a rate that is proportional to membrane and equilibrium potential. And if the concentration difference is known, then equilibrium potentials can be calculated. So across the membrane, net negative charges inside the cell are localized at the inner face of the membrane. Ions on one side of the phospholipid bilayer will interact electrostatically with ions on the other side. And negative charges inside the neuron and positive charges outside the neuron will tend to be mutually attracted to the cell membrane. So now we'll look at sodium. So sodium is 10 times more concentrated on the outside of the cell than the inside. So there's a large concentration difference, but electrically neutral. Again, we don't have any kind of door or any kind of opening. Now when sodium channels are open, and remember that we're selective only for sodium, ions are going to move down their concentration gradient because we have a lot of sodium on the outside versus the inside, but given the door, we will allow for sodium to flow through. This will leave negative charges behind. The electrical potential that balances out the concentration gradient, resulting in no more movement of sodium, is seen at positive 62 millivolts. And here we go with the same reasoning, that a net accumulation of positive charges on the inside and negative charge on the outside are going to retard the movement of the positively charged sodium ions from the outside to the inside. There's going to be an equilibrium that is going to be established, so there's no more net movement of ions across that membrane, thus resulting in a charge difference between the two sides. In the case of sodium, the inside of the cell is positively charged with respect to the outside. Now, each ion has its own equilibrium potential. So note when we were looking at equilibrium potentials, we were only looking at equilibrium potentials for one ion at a time. This equilibrium potential is the steady electrical potential that would occur if the membrane were only permeable to that ion. So we had the equilibrium potential of potassium, which was about negative 80 millivolts, and the equilibrium potential of sodium, which was about positive 62. Now, the Nernst equation calculates the exact value of the equilibrium potential for each ion in millivolts. Now, you're going to need to multiply your equation answer by 1,000 in order to get you into millivolts. The equilibrium potential is also called the reversal potential. Now, this takes into consideration the charge of the ion here the temperature in Kelvin, and the ratio of the external and internal ion concentrations. So what I would like you to do is work with me on this problem. Now for this class and for the test, what you will need is a calculator that is able to do logs or natural logs. If you're doing work with logarithms, you're going to need to actually put a constant out here, and I believe that's about 2.3. But here's the equation using natural log. So one question on your test might be, a hypothetical cell, X, has the following makeup for sodium at 37 degrees Celsius. Inside, 15, outside, 122. If you're asked to find the reversal potential, where do you even begin? So, I give you all this information. I give you all this. I give you R, T. I won't give you Z, because Z is something that you should know. Faraday's constant, and I give you the ionic concentrations. All you have to do is just plug it in. So we'll do this one together. So the equilibrium potential, or the reversal potential, for this cell that has permeability of, or a concentration of sodium inside at 15 and outside of 122. So let's say E, so equals R, which is 8.314, times temperature, so we're at 37. Remember that in order to produce this amount in Kelvin, you have to add 273 to this value. This is going to be divided by the charge of the ion, so positive 1, because sodium has a charge of positive 1, multiplied by Faraday's constant, which is 96,485. Now that value times the natural log of the concentration outside, which is 122, divided by 15, which is the concentration inside. So I'll do this on my calculator. 8.314 times 37 plus 273 divided by 96,485 times the natural log of 122 divided by 15. This will give me a value of 0.055988. Now, you'll probably be like, well, Stephen, that doesn't make sense. What you need to do is now multiply your value by 1,000. So this value right here is in volts. You need to get it in millivolts. So your answer now becomes, for this cell at 37 degrees Celsius, the reversal potential of sodium is now 55.988 millivolts. Now this is a cell that is only permeable to sodium. So at 37 degrees Celsius, with an inside concentration of 15 and an outside concentration of 122 millimolar, the reversal potential, or the point at which there is going to be a cessation of movement, is 55.988 millivolts. So not too bad, right? Just remember that you need to multiply that value by 1,000. Across the membrane, potassium is more concentrated on the inside. Sodium, chloride, and calcium are more concentrated on the outside. So this is just a brief chart that's given in your book of concentrations in millimolars on the inside versus the outside. So at 37 degrees Celsius, the equilibrium potential for each of the following ions is given here. Now note, this is only in the instance of permeability to that single ion channel. So in this case of a cell only being permeable to potassium, Negative 80 millivolts would be the equilibrium potential. Sodium, positive 62. Calcium, 123 millivolts. And chloride ions, negative 65 millivolts. This will be an important table for you to reference. So ionic concentration gradients are established by the actions of ion pumps. One of the best examples of this is the sodium-potassium pump. This is an enzyme that breaks down ATP. It's known as an ATPase when sodium is present. The chemical energy released drives the pump, exchanging 3 internal sodiums for 2 extracellular potassiums. And we'll revisit this sodium-potassium pump when we talk about the action potential. Now, what this pump does is it ensures potassium is concentrated inside the cell and sodium is concentrated outside the cell. Now, because you're going against concentration gradients, you have to expend metabolic energy. There are other examples of pumps in the nervous system, one of them being calcium pumps, which helps to actively transport calcium out of the cytosol. And without these ion pumps, the resting membrane potential wouldn't exist and the brain wouldn't function. You need these resting membrane potentials in order for neuronal communication to occur. If you didn't have these pumps, that could never be reestablished. Now, neurons are not permeable to just a single type of ion. You don't ever have a neuron that's only permeable to sodium. That's not what happens. You have relative ion permeabilities of the membrane at rest. So the neuron membrane is permeable, or it means that it is allowed to have different ions go in and out to more than one type of ion. So if it was only permeable to one ion, then that ion would flow until its membrane potential was equal to the equilibrium potential. The membrane permeability helps to determine the membrane potential. So equal permeability of potassium and sodium would give you some average of the equilibrium potential for potassium and sodium, right? If they were equally permeable. But we know that's not the case. So we know that there's a lot more potassium inside the cell. So the membrane potential is between sodium and potassium's equilibrium potential, but it's much closer to that of potassium. Now, using the Goldman equation, this takes into account permeability of the membrane to different ions. So the relative contribution of each ion depends on concentration gradient, so how much is in versus outside the cell, and permeability. So you take your gas constant multiplied by temperature in Kelvin, divided by Faraday's constant, multiplied by the natural log of the permeability of sodium times of the concentration of sodium outside, plus the permeability of potassium, and permeability typically being measured in meters per second, concentration. Now note here, because this will probably be a question on a test, because chloride has an opposite charge, in this case opposite of positive, you need to flip these terms. So here notice that it's sodium out, potassium out, chloride in. So if I were to give you all 3 of these ions and ask you to calculate a membrane potential, you would need to make sure that chloride in is on the top in the numerator versus the denominator. Okay. So, if relative permeabilities are known, it is possible to calculate the membrane potential at equilibrium using this equation. So, in terms of basic stuff, we'll stick with just sodium and potassium, but you should be aware that they're also permeable to chloride. And this is one of those instances where I'm going to tell you now that I'm putting chloride on your test. It's sort of tricky to some kids, and that's why I've left it out in the past when I've taught this class, just because, but in an online class where you get a weekend to do the test, you should be okay to do. Now, what I'll do is I'll give you about maybe like 2 minutes to work this out. I'm going to work it out on my paper right here. That way we can go through it together. Basically, this is all plug and chug. So You can either pause it or just sort of wait 2 minutes because I'll just let the video keep running for about 2 more minutes. Okay, so I've asked to calculate the membrane potential for the following hypothetical cell. This is going to be the potential at which there is going to be a net cessation of movement where you're going to have, based on the permeabilities of your given ions, potassium and sodium, based on those given permeabilities and where those ions are inside versus outside, this is going to be at resting state what your membrane potential is for this given neuron at a particular degree. So your membrane potential is equal to 8.314, which is the gas constant, multiplied by 37 plus 273, because that's how you get Kelvin, divided by Faraday's constant of 96,485, multiplied by the natural log multiplied by the natural log of the permeability of sodium, which is 1, times sodium out, which is 150, plus the permeability of potassium, which is 40, times potassium out, which is 5. And because you don't have chloride ions, you don't factor that in. So it's essentially plus 0. And then you have 1 times 15, and then 40 times 100. My recommendation is to do all of this inside the parentheses first. So I get the natural log of 350, which is this term, divided by 4,015. You should get, if doing this, you should get negative 0.06517. But remember that you need to multiply this whole equation by 1,000 to get your answer in millivolts. Now, remember for your test, I will most likely 99% give you permeabilities of chloride ions as well as concentrations of chloride in versus out. So remember that when doing that, you need to make sure that chloride goes on the top versus the bottom in terms of in versus out. So relative ion permeabilities at rest, there's a selective permeability of potassium channels. This is a key determinant of the resting membrane potential. There are many types of potassium channels that exist. Lily Yu-Nung John have looked at amino acid sequences in a family of potassium channels and have seen and studied multiple different types. One of these includes the Shaker potassium channel studied in the fruit fly Drosophila melanogaster. Potassium channels have 4 subunits right here. And the channel selectivity permeability to potassium ions is seen through this pore loop region, which helps to contribute to the selectivity filter, meaning that it's not going to let calcium ions flow through. It's going to be selective for potassium. Now, McKinnon in 2003 received a Nobel Prize for work on potassium channels. So seeing that scorpion toxin blocks the potassium channel by binding tightly to a site that is found within the actual pore. Now, this was used to identify the stretch of amino acids that helps to form the inside walls and the selectivity filter of the channel. This provides a physiological basis of ion selectivity. Now, mutations in specific potassium channels can disrupt neuronal functioning, and we see this in inherited neurological disorders or in channelopathies. So here is the overall protein structure of the channel pore. And this is looking at an electron microscope of a Shaker potassium channel in the cell membrane of a fruit fly in Drosophila. Now, regulating that external potassium concentration is vital for physiological functioning. The resting membrane potential is close to the equilibrium potential of potassium because it's mostly permeable to potassium. Regular neurons are mostly permeable to potassium, and there's a very, very, very high controlled environment for our sodium. And we'll get into that in the next video. But the neuron is mostly permeable to potassium. The membrane potential is sensitive to extracellular potassium, where increased levels of extracellular potassium have the ability to depolarize the cell membrane. Depolarization simply just means that there's a change in the membrane potential to a less negative or a more positive value. So here's the concentration of potassium that's found on the outside of the cell, and here's the membrane potential of a given neuron. You see that right around here, if we are greatly increasing this to right about greater than 10, this value goes up and up and up and up. This is bad because you need your membrane potential to be at a resting state in order to actually work correctly. We have that resting membrane potential of negative 70 millivolts where you have semi-leaky potassium channels and tightly gated sodium channels. But if you have a membrane potential of greater than negative 70 millivolts or even greater than the activation threshold, then your neuronal functioning is going to not be there. And that is the case in terms of the death penalty for lethal injection. And that's why it works. And I have a slide on that in a second. So mechanisms that help to regulate the external potassium concentration include the blood-brain barrier. And I have a good paper on that I've put on Blackboard. So if you choose to read that paper, it's a good topic. But the blood-brain barrier is a specialization of the walls of brain capillaries that helps to limit not only the movement of potassium, but other bloodborne substances into the extracellular fluid. And another one, which is another method that we talked about in the last lecture, was the potassium spatial buffering done by astrocytes, which are a type of glial cell. Now, the membrane potassium pumps that concentrate potassium in their cytosol, this helps to depolarize the astrocytic membrane. So inside the astrocyte, there are membrane potassium pumps that will bring that potassium into the astrocyte's cytosol. That potassium entry will increase the concentration of potassium inside, which will then dissipate over the large area across the astrocyte. Hence, why it's known as potassium spatial buffering. The large amount of potassium inside the astrocyte is able to be buffered out across the surface area of the astrocyte, taking away from the direct extracellular area that can impede with functioning of neurons. So implications of an extracellular increase in potassium outside the brain. Your muscle cells do not have the equivalent to the blood-brain barrier or glial buffering mechanisms such as seen as in astrocyte spatial buffering. Elevations in potassium in the blood can have serious consequences due to the facts of muscle cells. So lethal injection works this way. So Dr. Jack Kevorkian, aka Dr. Death, and his patient Janet Atkins, we have saline solutions, sedative to relax the patient, and potassium chloride and muscle relaxant mixtures fed through an IV. Now what happens is this potassium chloride, you have a lot of potassium ions that are flowing into the bloodstream. Cells need an appropriate resting potential in order to operate efficiently. The membrane of every cell is very sensitive to changes in the extracellular concentration of potassium. So if potassium rises in the periphery through all your muscle cells, then the resting potential of your cells becomes quite diminished. And we already talked about how how the concentration of potassium in the outside of the cell can have a direct impact on the membrane potential of the cell. So heart cells can no longer generate impulses due to the fact that their membrane potential is messed up due to the increase in extracellular potassium. This leads to contraction and cessation of action, thus death. So the activity of the sodium-potassium pump helps to maintain large potassium concentration gradients, as well as make sure that sodium and potassium are on the right side of the cell. We talked about the role of ion pumps, as well as the role of channel proteins. Potassium channels help to contribute to the resting potential, and at rest, their neuronal membrane is highly permeable to potassium. And we spent a lot of time talking about the electrical potential difference across the membrane being similar to a battery. The charge is maintained by the work of ion pumps, and we actually have the ability of current to flow when there is actual channels or doorways whereby our ions can flow through. In the next lecture, what we'll talk about is the action potential. So we're going to be using all of this information that we've already talked about to be able to say, okay, what happens when the resting membrane potential of negative 70 millivolts is depolarized? Meaning what happens when a neuron gets from negative 70 to negative 55, let's say, and fires an action potential. What happens during that whole process? So, if you need to, please watch this video over again. Feel free to take some notes, go through it and practice a few of those equations on your own. That way when you get to the test, you can feel comfortable with it. And I'll see you in the next video.